All right, we'll go ahead and get started. So this is our last presentation for the oral presentation session 1. So we have Melvin Soriano presenting on therapeutic cardio thread production for heart therapy.
Welcome everyone. Thank you for coming down to SCCUR 2019 in San Marcos. My name is Melvin Soriano, and I'm one of the researchers for the Cardiac Project, which is a part of the Central Coast Biotech Institute. And just a little bit of background. What we do at Central Coast Biotech Institute is we try and find different therapeutic solutions and biotechnical models to see how we can apply stem cells, more specifically differentiated cardiomyocytes. And a little bit of more background, this project was started 7 years ago by a group of undergraduate researchers at Oxnard College with the help of Dr. Jim Harbour that we do have in the crowd today. Thank you for coming today. And the project was founded on a desire to find a solution for a problem. And that problem is sweeping not only the United States, but just about every country in the world. And that problem is cardiovascular disease. Cardiovascular disease is the leading cause of mortality worldwide. And this is because tissue does not efficiently generate new cardiac cells with current therapies. Thus, if the heart cannot produce new cardiac tissue, then the heart will not be able to pump blood efficiently throughout the body, which can lead to death. That is why there's demand for heart repair therapies throughout the world. So here's just some statistical data that I gathered. An estimated 6.2 million Americans above the age of 20 have had heart failure from 2013 to 2016, and the estimated prevalence of heart failure is set to rise from way back 2012 to 2030. And this trend has been seen to hold true, as due to pollution, stress, and the increase of general obesity in the population, there has been an increase in heart failure. And also another reason that our group was very interested in trying to find a solution for this was that $30.7 billion was spent on heart failure and heart-related incidents alone in 2012. So to sort of emulate the conditions found in the human body, specifically the human heart, we decided to settle with the P19CL6 cell line. It's a cell line derived from a mouse with ovarian cancer, and we maintained it at 5% carbon dioxide environment at 37 degrees Celsius. And the reason why the P19CL6 cell line is so widely used is because it's only biosafety level 1, and it requires very low maintenance, if I could say that. Media only needs to be changed about twice a week, and the media that we use is alpha minimal essential medium supplemented with the following ingredients, most notably dimethyl sulfoxide, which is actually what we use to differentiate the stem cells into cardiomyocytes. And these cells are grown on tissue culture treated and non-treated surfaces, and in an ultra low attachment petri dish with covalently bonded hydrogel surfaces. And this hydrogel is proven to increase the beating in our cardiomyocyte structures. And to kickstart our sort of cardiac project, we decided to see how we could implement stem cells onto sutures, and we came up with the banjo model. Yeah, funny name. So we decided to suspend 5 parallel sutures across these two posts. And we would add cells onto those sutures drop by drop. And this replicates the conditions found in robotic-aided sinoatrial surgery, which is essentially the region that sends signals to the rest of the heart for it to contract and pump blood. And the chromic gut sutures that we use are really translucent under a microscope, so we were able to see our cells under the microscope. And as previously stated, the hydrogel enhanced cardiomyocyte activity. And the next model we decided to roll with is the meat Xenograft model. And in this model, we illustrated how bridging and complex macrostructures can occur in a xenograft. So for the meats right here, we decided to use pig heart tissue, and we decided to see how our mouse stem cells reacted or sort of coexisted in an environment with this pig heart tissue. Also, our design incorporated separate housing sections for different cardio tissue. So what we decided on is using these golden arches to keep the meat from interfering with the cells under the microscope. And our latest and greatest model, the D model, as in cardiac, is a model that incorporates the last two prior iterations and includes modifications to fit a pacemaker. So if you can see in this little area right here, where the sutures pass through, that is an area where we are able to drop down platinum wires that connect to a pacemaker and conduct an electric current from one side to another. And this model is basically an all-in-one, so it allows us to test out the previous experiments separately or all together. And so now we have the D model with the pacemaker, and to sort of connect the pacemaker from the outside to the in vitro model, we decided to modify a 6-well Petri dish to have holes and these little guides to have the platinum wires drop down into the model like such and conduct the electric current. We also decided to add signs, so there's a plus sign, there's a minus sign, that corresponds with the minus signs and plus signs on the lid to make it easier to figure out where to place the cathodes and the anodes from the pacemaker. And this is the conceptual design of a temporary pacemaker. So the anode and the cathode wires are placed in close proximity to each other, and this current stimulates cardio tissue for lateral movement of cells. The goal of a temporary pacemaker is to regulate beating in a region to match that of a heart. And not only do we use this in our cell model, we also use this to initiate the beating of our cardiomyocytes and also add direction as they grow and differentiate into cardiac tissue. So this is a little diagram that we came up with. And so this idea of adding cells, cardiomyocytes, onto the suture, we called the cardio thread. And it's essentially a little machine that takes a patient's cells at bedside and weaves them into a suture that can then be used during and post-surgery to help regenerate cardiac tissue. And so these little strings will have the transcription factors to differentiate the stem cells. Oh, and also we have a little pacemaker setup here that while the cells are cultivating, we can add that pacemaker to add direction and initiate beating. And the machine that we came up with to add the stem cells more fluidly to the suture is the cardiac assembler. And this cardiac assembler, there's essentially a preloaded sterile suture right here, and it goes under these two discs and dips into these troughs that contain different solutions that our sutures will be coated in. So I know there's 5, there's supposed to be 4, and the wells contain fibrin, gel cells, and thrombin. And what we do is you just turn the knobs and it rotates the suture, one section that can then be dipped from one trough to the other. And you may be wondering, what were those little bobbins right there? Well, they are the suture models. These are models that we designed to help cultivate this sort of cardio thread in vitro. And right here, we just have a plain, simple bobbin to house our sterile sutures. And we also have this more complex model that allows us to translate the sterile suture to that bobbin, ready for cultivation. So this suture would be, the needle of the suture would be placed in this scaffold and would be spun around and tied at the end in these posts. And we also have these guide rails for platinum wires to drop down into the model and conduct an electric current. And we have a second model because we didn't like the first one, because we found it very inefficient to rotate one section, dip, 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 then rotate one section, dip, dip, dip. So we decided on the cardiac assemble model 2. And this is essentially a continuous assembler that allows us to fluidly transfer the suture throughout all the troughs in one single motion. And we also added this mechanism that allows for us to turn the knobs and the knobs not go back that allows the suture not to go backwards, only go forwards. And the applications, adult human heart has poor regeneration rate, and the pacemaker model addresses concerns during and after surgery. And so what the cardio thread could be used for is atrioventricular septal defects, atrial septal defects, and complete atrioventricular canal defects. That was a mouthful. And these are essentially diseases prevalent in heart failure that would lead to heart failure. And these are our references. Thank you.
Thank you very much. That was a great presentation. So any questions from the audience?
Yes.
So what is the purpose of you using the fetal bovine serum in your media?
Fetal bovine is essentially a growth hormone for the stem cells to take about like 3 to 4 weeks for them to fully grow and contract.
So the growth hormone is in the serum?
Mm-hmm.
Do you know what growth hormone it is?
Growth hormone. I wasn't talking to my lab. My lab discussed this when I was not present. So I can get you that information later on, but not right now. So please come talk to me afterwards.
Do you have any plans for like in vivo experiments or any future
Yeah, way
outlooks
down
for
the
that?
line. Right now, we have yet to test out this bad boy. So once we get that settled out and get the pacemaker running through that spindle model, I think that'd be the next step.
Does the spindle model, would that be outside? So if you're testing it on like a rat or mouse per se, how would you get that needle and the sutures like into like the meat per se?
Of the mouse?
Yeah, of the mouse.
So we could actually unscaffold or unload the fully cultivated cardio thread and use that to stitch up the mouse and see how the cells interact with the mouse tissue. Yeah.
So what, for the experiments where the 3D printed material is touching the cells, what is the material made of?
Oh, yes. So I work heavily in this department. So at Oxnard College, we have a STEM center that has a ton of 3D printers. And the material used is PLA, which stands for polylactic acid. And it's a very biochemically inert substance that is safe for the cells to grow on. And we've been using that for the past 7 years. Any more questions?
Any additional questions? Thank you very much.